Okay. So this paper was about molecular testing for acute myeloid leukemia. So acute myeloid leukemia is a type of cancer that affects the blood and bone marrow and is characterized by a proliferation of abnormal myeloid cells. And AML can be classified into two different categories based on the genetic abnormalities that are observed. So the first one is those with chromosomal rearrangements that lead to translocation and fusion genes. And then the other one is those characterized by gene mutations, such as point mutations, deletions, and insertions. And in recognizing these two, it's important to recognize the molecular changes for AML so that we can classify and diagnose and look at the risk and treatment plans for um, various patients and their unique cases. And molecular testing can be especially helpful in identifying translocations and identifying specific forms of leukemia to offer specific treatment options and to identify certain recurrent gene mutations that are critical prognostic indicators and guides for treatments in the unique cases of AML patients. Um, so molecular testing broadly is any testing that reveals DNA changes, and more specifically, it's any testing that reveals changes in the nucleotide and DNA and RNA in a DNA and RNA sequence. And there are many types of molecular testing, but the ones that were covered were karyotyping, FISH, PCR testing, and next generation sequencing. So some of the types of testing, first, karyotyping is used for cytogenetic assessment of different diseases, and it basically involves analyzing the morphology of specifically metaphase chromosomes, so they can only be in that state. And then it helps us identify recurrent translocations and complex chromosomal changes that are indicative of specific types of AML, but there are some limitations. The cells have to be in their metaphase so to be analyzed, otherwise they can't be analyzed. And cryptic cryptic chromosomal chains are really like small ones, cannot be detected via karyotyping. So FISH, this is used to detect specific DNA sequences, which is different from cytogenetic. So DNA sequences on chromosomes, and it employs fluorescently labeled DNA probes to bind to complementary DNA sequences in the sample. And it's very similar to karyotyping in that it can be used to identify the chromosomal abnormalities that are associated with various AML, but it has a few advantages to um, karyotyping. Particularly, it's useful for detecting cryptic translocations that may not be visible with regular karyotyping. It can be applied to both metaphase chromosomes and interphase nuclei, offering a quicker turnaround time, and also like more samples can be used in analysis. And FISH is also a lot more sensitive than karyotyping and is widely used in the diagnosis of disease where rapid and accurate detection of the translocation is critical for timely treatment. And then PCR-associated molecular testing. It takes advantage of PCR amplification to make more copies of the specific DNA and RNA samples. And thus, this method is highly sensitive because it specifically targets genomic regions and uses short and precise primers. And PCR testing can identify specific mutations, insertions, deletions, and translocations with high resolution. One drawback is that some translocations may have numerous breakpoints, so many pairs of primers maybe have to be designed for these breakpoints, which can make it less practical for certain types of translocations. And then traditional sequencing is used to determine the order of nucleotides in a DNA molecule, and it is used in AML assessment to identify mutations such as point mutations, insertions, and deletions. The most popular one is Sanger sequencing, and it has a detection limit of about 15% allele frequency, meaning it cannot detect many mutations that are present at lower frequency. Pyrosequencing is another kind and can be more sensitive and easier for quantifying variant allelic frequency, but it typically analyzes shorter DNA sequences, so it's not good for a larger scale analysis. And then next generation sequencing, this is a DNA sequencing technology that allows for rapid sequencing of large stretches of DNA pairs. So this one can be applied large scale as opposed to the other one. So yeah, it can be scaled largely, or it can be used for smaller projects like targeted sequencing of specific gene panels. NGS is really sensitive, so it can detect low allelic frequencies, so it's suitable for detecting minimal residual disease. And it has many applications in treating AML. It can be used for disease diagnosis, treatment decisions, monitoring of the disease, and single-cell sequencing. Some other technologies that were mentioned were spatial transcriptomics. It combines molecular tests with microscopic location to observe molecular changes within specific anatomic structures of cells. And then circulating cell-free tumor DNA testing used for detecting tumors in the tumor DNA in the bloodstream, which can be useful when primary tumor tissue is difficult to obtain, and is also helpful for monitoring treatment and response or disease progression. And then the circulating tumor cell test is a technique for identifying tumor cells in the bloodstream and is particularly useful for predicting tumor metastasis or recurrence. So here's some examples of AML mutation testing. So the first mutation is APL. It's characterized by T1517 translocation, resulting in PML-RAR fusion gene. It accounts for 5 to 8% of all AML cases and is associated with the disseminated intravascular coagulation. This mutation has a significant death rate due to the coagulopathy, but responds well to tritoin treatment, leading to a favorable prognosis if it is diagnosed. Thus, prompt and correct diagnosis is critical for effective treatment. Karyotyping can detect most of these translocations, but may miss cryptic translocations and has a longer turnaround time. FISH assays can detect translocations more quickly and are sensitive to cryptic translocation, which is really important in early diagnosis of this mutation. And PCR assays are designed to detect this translocation and are useful for monitoring the minimal residual disease. Alright, AML. So the next mutation is AML with a T1821 translocation. So this translocation results in RUNX1 T1 fusion gene. AML with T821 generally has a high rate of complete remission and favorable outcomes when it is diagnosed. And in terms of detection, it can be detected via karyotyping, but cryptic translocations may result in false negatives. FISH assays on interface cells can detect some cryptic translocations, offering a greater sensitivity than karyotyping. In cases with less than 20% blast, the presence of T821 translocation still supports the diagnosis of AML. The next mutation is AML with a CEBPA mutation. This is seen in about 4 to 9% of young AML patients, and it is a 1 exon gene with mutations occurring throughout the gene, making sequencing assays suitable for detection of this mutation. Sanger sequencing is the most popular to detect this mutation, but has a lower detection limit of about 15 to 20% varying allele frequency. NGS can also be good for detecting this mutation. Hybridization capture NGS assays are more effective than amplicon assays due to high GC content in this mutation. And this mutation is considered a low-risk group with favorable outcomes. AML with the FLT3 mutation. This is found in approximately 30% of AML cases, um, and however, it is associated with an unfavorable prognosis. FLT3 inhibitors are available for treating AML with FLT3 mutations. Detection methods for FLT3 mutations include PCR with fragment analysis and NGS assays. The PCR fragment analysis calculates the ratio of FLT3 IgT to wild-type FLT3, which is used for risk stratification for the patient, and then NGS assays can detect FLT3 TKD mutations effectively, but FLT3 IgD detection can be challenging due to variable insertion sizes. Hybridization capture at NGS may be more suitable for detecting FLT3 IgD mutations than amplicon NGS. And then the next mutation is MRD. So MRD in AML refers to the presence of leukemia cells at the level that is listed. Both flow cytometry and molecular tests are used for MRD measurements. Certain gene mutations like NPM1 and fusion genes like RUNX1 and RUNX1T1, or all the listed ones, are valuable for MRD tests due to their prognostic value. Some mutations are not suitable for MRD markers as they occur in healthy aging individuals and lack prognostic correlation. Real-time quantitative PCR is commonly used for single-gene MRD markers, while NGS is used for multiple gene markers. Unique molecular barcode technology in NGS reduces error and noise, enhancing its application in MRD testing. And the definition of MRD is expected to be more refined when there's more data availability, so this one is less common. And then finally, we have clonal evolution. This is like a well-known phenomena, and NGS assays are effective for gathering information on clonal evolution in patients. Single-cell NGS provides detailed insights into clonal evolution, and new technologies are enabling better personalized precision medicine by revealing clonal dynamics in AML. Yeah, I think that's it. Thank you.